To Quick and Dirty Med School. Today we'll be starting with histology. So this is the preparation for the paraffin slides. Once again, you don't really need to commit this to memory, but it's a good idea to know what exactly is going on to prepare for a slide, right? So you get a sample from a biopsy or a necropsy about 1 cubic centimeter in size, and then you fix it with formalin, which is a derivative of formaldehyde to preserve the structures and prevent the amine groups of whatever proteins from degrading. It also preserves the architecture of the tissue. Now, next you freeze to preserve the lipids, but that's kind of up to the discretion of what you're trying to test for, because in the next two steps, 4 and 5, those tend to remove the lipids and other, fat soluble molecules from the cell. So after 4 and 5, you infiltrate or embed the sample with paraffin or epoxy to stabilize the sample. Epoxy will cover in the next couple slides, but for a light microscope we use paraffin. Next we trim with a microtome. A microtome is basically a very, very, very precise paper cutter, but for cells. It cuts within 5 to 10 micrometers, and next we mount it and we rehydrate it and stain it with whatever stain that we need. Let's get to it. So the most common stain is hematoxylin and eosine. So hematoxylin is a basic dye, and it will stain acidic molecules like DNA, RNA, mitochondria quite strongly, and it stains in blue. Eosine is its counterpart. It's an acidic dye, and it will stain, collagen, elastin, proteins, whatever, pink. Let's see. As you can see here, hematoxylin and eosine by themselves are not very useful because they don't differentiate cellular structures and substructures very well at all. They kind of stain indiscriminately. So together, they produce something that is much more useful, like so. And it is because hematoxylin is metachromasia. What does that mean? It means that it's polyanionic, and that is pH dependent. That is because each organelle, structure, molecule has a slightly different pH, right? And hematoxylin, depending on what proton concentration is available, will stain deeper. For example, the nucleus here stains quite deeply because it is quite acidic. So hematoxylin will bind and stain deeper. So mast cells, cartilage, mucin, amyloids are acidic, and they will stain purple instead of blue. Next, light microscope. You don't really need to know all the parts of the microscope. Just in general, know that they're bright field. You use a light and whatever stain. And for fluorescence, you use UV light and a fluorescent marker. And all of these tissues and samples are oil immersion. That means that the tissue is submerged in oil between the slides and the lens. Okay, so moving on to the lipid special stains. Like I said before, they have to have a special stain because in preparation, these can be washed away. So to prevent that, we stain it with something that binds very strongly to them. The first is oil red O, and that will bind to the lipids and stain it red, as you can see here. And the nuclei will be stained black. So oil red O is quite useful because it detects fat emboli in lung tissue. It also identifies tumors such as lipomas and liposarcomas. Sudan black 4 also stains lipids, and it stains it black. Those two could be used to identify like Paget's disease. You'll study that more later on, but Paget's disease is disease of the breast, and the breast has a lot of adipose tissue, so that kind of like can give an indication that hey, maybe there might be some carcinoma might be happening, or there's some abnormal adipose growth here. And it also helps in detecting the glycogen storage diseases, because once again, the glycogen is like a lipid. So more lipid stains. Osmium tetroxide will stain lipids and myelin. As you can see, it stains the myelin, this kind of gross brown color. And PAS and Alcyon blue is very, very useful. It's one of the more distinct and specific stain there are. PAS is the periodic acid shift that is used to stain like carbohydrates and macromolecules such as glycogen, glycoproteins, proteoglycans. And PAS is always used with Alcyon blue because, like hematoxylin and eosin, these two are not very useful by themselves. So together they provide a counter stain. The PAS is going to be staining like acidic and neutral elements. So here you can see the goblet cells are stained quite deeply magenta because goblet cells secrete acid, and that is what PAS is binding to. And the Alcyon blue will stain like other mucins blue, but they don't stain the neutral mucins. Alcyon blue is a good combination that is generally used in kind of like intestinal goblet cells. Why are these useful? So these PAS and Alcyon blue is really used to identify cystic fibrosis and adenocarcinomas because in these two diseases they have increased mucin production. It also helps identify Whipple's disease, and it's a disease that affects the GI tract and cause malabsorption because the PAS will stain the macrophages quite well. They also stain the erythroleukemia, which is leukemia of immature red blood cells, and those cells will stain a bright fuchsia. All right, next. Okay, so connective tissue staining. There's a couple stains. The first one is the Mallory trichrome stain, and that differentiates the collagen and the smooth muscle from the cytoplasm. So the collagen will be stained this nice blue, and then smooth muscle is going to be stained red. And the elastic fibers you can barely see, but those are going to be pink. The Mason's trichrome. Let's go back to Mallory for a little bit. So why do we need to differentiate between collagen and smooth muscle? It's because in certain organs they should only have smooth muscles, but after disease process or damage, it will scar over and produce collagen instead, such as liver cirrhosis. So we can use the Mallory trichrome to identify any sort of trauma to structures such as the liver or the kidneys, because those can also scar over as well. So Mason's trichrome differentiates between the cellular from the extracellular products. So collagen is, as always, extracellular, and those will be stained green, and the cytoplasm is going to be stained pink. All right, Van Gissen's stain stains elastin and collagen. So this is particularly useful because you can see any tumor growth, It demonstrates the increase of collagen in those diseases, in those tumors. As you can see here, oh, you can barely see, but the cytoplasm right here is going to be yellow. The elastic fibers are going to be black, and the collagen is going to be red. So the next connective tissue is reticular fibers, and reticular fibers are very, very fine. As you can see, you can't even tell that they were here. So we use silver salts to make them show up, and they show up as very, very fine black threads. And we use this particular staining to see components of, the liver or the lymphoid organs, because they have a lot of reticular fibers. The next stain is Golgis method. So Golgis method stains not just the neurons, they also stain any kind of cell that conducts electricity. So it will also stain Purkinje fibers. And those show up as black over like a yellow-orange background. The Gemsa stain is one of the most useful stains out of all of these stains because it stains the peripheral blood cells. It stains, like, as you see, the nuclei like blue to violet, then the erythrocytes pink, and as you can see here, they also stain the platelets blue. So this is used for the diagnosis of like malaria, to see the Plasmodium species, babesiosis, the sleeping sickness, and schistosoma species. And in these, like, those are parasites, but in these particular diseases, these parasites will infiltrate the cells, and with this stain you can tell quite well what exactly happened. These diseases are particular, like they target like either neutrophils or erythrocytes, but you'll learn about that in more like physiology. Next, electron microscope. It is useful, but I highly doubt that any of you, if you're just like a plain old med student, is really going to encounter this, because this is really for research purposes. In practical medicine, I truly doubt you're ever going to see any of this. What do you need to know? You fix it with glutaraldehyde and osmium tetroxide, and you embed in epoxy, not in paraffin, because in paraffin you have to use a little bit of heat, and heat can denature some proteins and denature some enzymes. That's the more important part. You mount it on the grid, and then you stain it with uranyl acetate and lead citrate. The lead citrate will bind to the RNA, and the uranyl acetate will stain like membrane structures. And it's sprayed with something ridiculously expensive like gold or platinum, which is why the average physician really isn't going to use it. But as you can see here, it shows some incredible details. Like, you can see the receptors here, the glycocalyx here. You can even see, like, here, this, I think this is the flagella. So you do see quite a lot of detail, but more useful for research, less useful for a clinician. Same kind of goes with the scanning electron microscope and the scanning electron microscope only shows you a 3D outer surface. The transmission electron microscope shows you the insides, so it will show you the electrodense regions, which show up black, and the electrolucent regions, that show up white. They are useful to see like very minute details, organelles. With the transmission emission electron microscope, you can even see like the inner mitochondrial matrix and whatnot, but not particularly useful, like I said. There's some complications with whatever like preparation slides that you can have, because artifacts are They are distortions of the tissues. They shrink. There's wrinkles, spaces, precipitates form from the stain. Magnification, it's very limited. If you're using light microscope, like there's only so much magnification a lens can provide. And you have to keep in mind that cells are 3D, not just 2D, but looking at the slides, it can seem like only 2D or give a false appearance of 3D. And you have to know like the different orientations of the structures in the tissue, so that can also cause variation in their appearance. Let me see if I can go back a little bit. I forgot to mention the cryofracture is useful for these because it preserves the lipid membrane. It preserves the glycoproteins and whatnot that are in it, and it splits it right down the middle, so you can see both the inner and outer leaflets. Right. Photography is pretty useful. More for research, like I said. So you use an X-ray to find newly synthesized macromolecules, and you use it to detect radiation from labeled molecules. And these are used to see the synthesis, migration, division, performance, and like site of whatever drug that has been administered. Cultured, you should know this just so you know what exactly like a certain study is researching. So everything's a buffered medium, regulated for growth. Primary is the cells isolated from the tissues. Subculture is like the cells that are just moved to the petri dish. Cell line is the subclone after the first subculture. And the induced transformation for immortality is some kind of gene editing, some modification, probably some splicing, and that is to, that is for the cell strain. So the cell strain is to isolate like a subpopulation of a cell line that we want to continue research on. So we choose this after it's been positively selected from the culture, and we induce the transformation to it. So it will be immortal, so we can have like a germline. So fibroblasts are elongated, and they will attach to the substrate, which means they will attach directly to like the culture. Epithelial cells, they will attach in patches. Lymphoblasts are spherical, and they will float without attaching to the culture. I mean, not of course, not like float above the culture or anything, but they don't attach as strongly. Histochemistry is particularly useful because this targets enzymes, and enzymes are very, very specific, and nothing's good enough for them. pH has to be perfect, enzyme has to be perfect, the ligand has to be perfect. Otherwise, else they will degrade or just, not work. So you localize the cell structure using specific enzymes, and these will only bind, if the conditions are just right. So the cells, in order to preserve those conditions, must be frozen. A marker is introduced, and it will bind to our target of interest. And this is useful for drug screening and the development of medication. Oh, and that's it. Thank you for your attention, and good luck studying.